Jana Stoudemire. As the Commercial Innovation Officer at Space Tango, Jana Stoudemire leads the commercial market creation for biomedical and technology manufacturing applications in low Earth orbit. Today, you'll hear about Space Tango's work to expand capabilities and partnerships for stem cell discovery and manufacturing applications in microgravity. Go ahead, Jana.
Good morning, Liz, and good morning, everyone. Thank you so much for this opportunity to present to the group today. And thank you, Dr. Zubair, for an excellent opening presentation. I'm looking forward to hearing more from Dr. Sharma and from Dr. Shepard and Dr. Lin later on from CERM as well. I want to start by just saying, you know, it's really an exciting time, both in space and on the ground for regenerative medicine. And so I'm going to walk you through this morning a little bit of capabilities development that we're doing to support more sophisticated biology that really will help us to understand that regenerative medicine opportunity in microgravity, as well as some of the changes that are taking place on the space side that are helping us as well. So at Space Tango, our focus, as you probably know, I think most of you are aware of the work that Space Tango is doing, but we're really looking to inspire, innovate, and create a better future for humanity, utilizing the unique environment of space. And our mission is really to manufacture health and technology products in space that create value and transformational solutions for humanity. So we're very focused on opportunities to not only understand the fundamental science, but to look at how we can build a robust commercial space economy that has a new market segment in it for biomedical products. In terms of the automated systems, so we put together facilities that really support iterative R&D and manufacturing on the International Space Station. And part of the focus for us is to be looking at sort of, you know, more than one opportunity to be able to understand some of the complicated biology. It's complicated on the ground, and it's even more complicated to sort out in space, as you were just hearing from Dr. Zubair. Just briefly, I think everyone probably knows we're located, our headquarters are in Lexington, Kentucky. I'm actually based in San Diego, California. We have also part of our team in Washington, D.C. for strategic engagement with our congressional representatives, and we have ground operations in our Houston facility, satellite facility in Houston, Texas. Launch operations are performed out of NASA Kennedy Space Center at Cape Canaveral and Wallops Flight Facility, Wallops Island, Virginia. And on ISS, we actually have our 2 Tango Lab facilities and our power descent utility locker, which I'm going to talk a little bit more about in this presentation, but it's a newer capability that has been developed by Space Tango to allow us to have power on ascent so that we can actually maintain the biology as we transit to the International Space Station, as well as while we're on orbit. So you heard a little bit about rockets, and I know that we talked, that Dr. Zubair mentioned the splashdown. Just for background here, for anyone who is not aware, there are 2 providers for commercial resupply missions, which is where the science that goes up actually flies in. One is SpaceX, the Falcon 9 and the Dragon capsule, which everyone I think has seen quite recently, even with American astronauts on American rockets from American soil, which is fantastic. And Northrop Grumman also has the Cygnus capsule, the Antares rocket and the Cygnus capsule. Just a little bit of background here for anyone who is not familiar with these vehicles and space. The Dragon capsule is the only one that returns back to Earth with samples in it. The Cygnus vehicle goes to the ISS, delivers science and supplies, but actually is the trash vehicle that is designed to burn up on re-entry. So when we talk about biological payloads, and you know, Dr. Zubair mentioned, SpaceX is really the only capsule where those samples can return. So often we try to fly biological payloads on a SpaceX mission because they can go up and back in the same vehicle. You can actually go up on a Cygnus vehicle and come back on a Dragon, but there's a little bit of time between those launches, and so the sensitivity of these payloads we try as much as we can to put them in the same mission. The exciting part of what's happening on the space side is that in addition to those 2 vehicles, we are now having new vehicles come online and new opportunities for flights. So where it's been difficult and sometimes a long time frame to be able to get to an opportunity for launch, these additional vehicles and providers will be able to give us an opportunity to really have the increased access that we need. There's also some future commercial facilities that are being developed. So ISS, as everyone knows, this is the 20th anniversary year. It's been in orbit for quite a while as a laboratory, but recently NASA actually awarded the docking port to Axiom Space for a commercial space station that will be used to really maintain our presence in low Earth orbit once the ISS is decommissioned. Space Tango is actually working also on our own free flyer, an automated facility that we will be able to use for manufacturing on orbit of both technology and biomedical products. But it will give us the ability to really be on a schedule that will be orbiting, we won't be docking, there won't be people on there, so we can kind of control the timeframes of up and back in microgravity. So that's a little bit about capabilities, and it's good to see the increased access. But I want to switch gears a little bit and talk now about what it looks like to do research in space and what kinds of capabilities exist. So I thought I would start first with just sort of the basic overview for an understanding of what a lab actually looks like in space. On the top here, you see your typical lab bench with everything out on it and lots of bottles and reagents and all kinds of equipment. On the bottom, you see the inside of the ISS. Everything is bolted to a wall or put into a locker format, obviously so that it doesn't float away. And this really is one of the big differences about working in space versus working on the ground. Simple things that you would do in the laboratory, like a media exchange, that would take maybe 45 minutes, in space manually takes several hours. So it's really a very different process. Our facilities on ISS, we have 2 Tango Lab facilities, as I mentioned. The first one was actually installed in 2016, and the second facility was actually put up in 2017. So these facilities give us the ability to collect data from payloads that are automated and remotely control those payloads from the ground. It removes the astronaut hands and time that is very expensive and can at times be difficult to manage. The crew's schedule, as you may be aware, is very busy while they're on ISS, not only the research that they're doing, but the work that they're doing on station and the exercise that they need to do just to maintain their own physiology. So when we think about ways that we can help to accelerate that science, automating it and taking astronaut hands out of the process is something that we see can really help. The Power Descent Utility Locker, which is shown in the bottom picture here, is the locker that I mentioned at the beginning that we created to help us on ascent really control the conditions that you need to maintain cell culture on the way to the ISS. And once this facility is installed on board, we can downlink the data that we retrieved during the ascent of the vehicle and actually continue the experiment. Just a brief mention here of the facilities and sort of the flexibility. You know, one of the things that we also did with our Tango Lab facility was to take it out of its locker position and put it into a docked capsule, the Cygnus capsule, while it was docked to the International Space Station, really showing that we can kind of extend the laboratory that exists. Space is at a premium on ISS, as you can tell from the pictures. When you see everything, you know, every space is used. So to have the opportunity to be able to extend the laboratory facilities to a vehicle that's docked to station also increases our capability. This is just a short video of what the actual uninstall. These are 2 of our Cube Lab automated facilities, which I'll give you a little more detail on in just a minute. that are being removed from that Paul facility and put into cold storage. So each of these cube labs contain, this is one of our tissue chip payloads for the NIH, and each of these cube labs contain science that has been now conducted, long duration cell culture on station and now being moved into cold storage for return. So a little bit about what's inside the cube labs. And as I mentioned before, the automation for us is something that we understand to be able to support the sophisticated biology that is now a focus of regenerative medicine. We really need to have systems that allow us to look at different configurations of cells that can be grown in an automated way. So the cube labs that we've designed they permit long duration cell culture for things like tissue chips. We have 6, 12, and 96 well plate configurations, as well as bag configurations. And we've added to the cube lab an automated microscopy system. This is a great group from San Diego, and I have to sort of give a shout out to San Diego because they are local, but Etaluma is a company that actually have optics from Etaluma. It's a perfect scope for ISS because of the fact that it is not only very compact and can fit into the cube lab, but it also has a unique vibration resistance. And so we're really excited about increasing capabilities to do on-orbit measurements with a scope like this in an automated way. So let's talk a little bit about translating ground science to space science. And when I talk to the teams that we work with, I always talk about the fact that it's not really just the translation of ground science to space science. In our case, it's the translation of ground science to automation and then to space science. And there's lots of things that we need to think about. So the conditions on ascent, for example. What's the metabolic state of your cells, and how do you want them to arrive at the ISS so that you can really make sure that the data that you're capturing is data from a microgravity environment. The conditions on orbit, the timing for evaluating the desired effects of microgravity that you're looking for. So media samples and reagent storage, temperatures that need to be maintained for conditioned media that has growth factors in them within the same automated system as an incubator that keeps the temperature of the cells at 37 degrees. And then, of course, the sampling and imaging time points and how we can download that data and look at that during the course of the cell culture that goes on orbit so that we're able to make any adjustments that might be needed in near real time. And then the return of your cells. So fixed, preserved, frozen, live, those are all capabilities that we have. And in conjunction with doing on-orbit measurements, we want to make sure that we bring back cells that allow some additional post-flight analysis as well. In terms of the cell culture capabilities that the automated hardware we're developing is designed to provide, so NASA safety certified, obviously the systems that go up on ISS are at least 2 levels of containment. They're all built for biocompatibility and designed for sterilization of the components before the flight build. They have the ability to control temperatures, standard conditions for cell culture, 37 degrees, 5% CO2, do automated media exchange, media sampling over a time course, imaging, drug challenge, if that's something that you want to look at as well. And then obviously the fixation and cryopreservation or live return of cells. We've also, because of COVID-19, developed some additional benchtop capabilities. Everybody knows it's a little bit difficult to be in the lab right now. And those that are in the lab, for us to be able to visit and sort of be right there with some of the scientists, that's been a little challenging under the current travel restrictions as well. So what we've done is put together a benchtop capability that really allows the basic sort of cell feeding and agitation that you would see within the cube lab while it's on orbit to be remotely controlled from the ground. So we have a web-based GUI for our partners to control and for us to control from Space Tango at our remote access points. And we have data that we can pull from that and put into a portal that we're able to share with the researchers, and we can all watch as we're working through the validation of these systems to make sure that they're working the way they need to. So the focus on automation for us, there's a lot of reasons why. You know, in addition to the fact that astronaut time is expensive and sometimes difficult, you know, to make a change on a near real time is often a little tricky and challenging because the crew schedules are so full, but also for the consistency. You know, we're very lucky to have people like Peggy Whitson, and you're going to hear from Dr. Sharma next, I think, about the work that was done by Kate Rubens, which is phenomenal. And someone like Kate Rubens is an amazing biologist here on the ground, but not every astronaut that is on station is Peggy Whitson or Kate Rubens. And so we feel like the automated systems not only give us the ability to have higher throughput, but also the improved consistency. And we really focus a lot on making sure that those cube labs really have temperature, pressure, gas concentration, pumping speed, imaging, and other types of sensors that standardize the science environment. We take a step-by-step approach to product development. The automation allows for on-orbit tests of sub-technologies, so we really will look at each piece of the different part of the payload that's going to be automated, and sometimes we actually even look at that in a microgravity environment with things like beads or other ways to simulate before we actually put cells in and fly it. There's never enough testing that can be done on the ground to get ready for these types of experiments. The translation, as I mentioned, from bench to automation really does require that we grow the cells in the system and make sure that not only the engineering design meets the requirements, but that the biological requirements are met as well. And the ability for the near real-time data monitoring that I mentioned, you know, downlinking data to our remote station allows us to have the ability to even do things like make changes to the ground controls. So we have options for doing either parallel controls with a slight offset so that we can pull that data from what's happening on orbit and make adjustments if we need to, or at times some payloads actually an asynchronous control, you know, done after the work is done on station to match those conditions exactly, you know, makes sense. Most of the biological payloads, obviously you want to load them with the same cells that you're flying so that you limit the variability there, but there are opportunities to look at controls kind of from both perspectives. So now you know a little bit about what the hardware looks like. What happens when we actually now have tested the system and are ready to fly? We have pre-flight loading that goes on at Kennedy Space Center. So most of our teams will come down and be with us for several days before the launch, getting ready. And as anyone who has flown knows, launches are always called launch attempts because they can sometimes move. And with biological payloads, you really need to be prepared to have other sets of cells that are available to you in case there's a launch slip. Once the actual pre-flight loading is completed, the payload is then turned over to NASA. It is loaded into the rocket and actually sent to the International Space Station. Once the vehicle docks, it's unloaded, and the cube labs are then attached to payload cards that give us that remote capability to control and see what's happening on the ground while we're on orbit, and loaded into the Tango Lab facilities. Once the ground control operations and the cell culture period is completed, then the reverse happens. The cube labs, as you saw in that video, are actually detached and, you know, returned to us in the vehicle, and then splashdown, payload return. As Dr. Zubair mentioned, you know, we were picking payloads up since I'm here in California, right at Long Beach, and now this year we're going to actually be in the Atlantic, so closer to the Kennedy Space Center, where there are facilities to actually take your payloads and samples and right at return into those same facilities where you loaded previously. Just briefly, so we've 17 missions so far since our Tango labs were installed in 2016 and 2017, 88 payloads and 156 experiments. And I'm just going to briefly give you an overview of the types of projects that we're working on right now. So I mentioned NIH Tissue Chips in space. We have a partnership with Emulate doing a blood-brain barrier chip. We also, in the Tissue Chips in Space 2.0, are working with Emulate on a gut chip. And we also have two other projects, one with University of Pennsylvania and Children's Hospital of Philadelphia for bone marrow and lung, and one with University of Florida, Gainesville for skeletal muscle. Really excited. These payloads are coming up on SpaceX 21. So we're going to be very excited to see a lot of this work starting to bring data back to us as well. In terms of the NSF Tissue Engineering collaboration that CASES has put together with NSF, we have 3 payloads that we are actually supporting. One from Mount Sinai on cardiac stiffness, one from University of Michigan on mechanotransduction of osteoblasts, and then one from University of Texas and Texas Tech that is actually looking at cardiac organoids. So we're very excited to be working on several of the projects with NIH and NSF. And recently, we also flew brain organoid models, which you may have seen some information on these payloads as well. We're very excited to work with National Stem Cell Foundation and also with University of California and Arthur C. Clarke Center for Human Imagination. These 2 organoid payloads, one actually looked at neurodevelopmental biology, so very early stage organoids, and the other one looked at neurodegenerative disease using iPS cells from patients with Parkinson's and primary progressive MS. So in addition to the more 2-dimensional tissue chip models, moving up to 3-dimensional stem cell models is something that, you know, again, takes the biology to yet another level of sophistication. And recently, in April of this year, we actually were very pleased to be awarded by NASA 3 projects that are really focused on commercialization for regenerative medicine. You know, the support of the public-private partnerships is really what will drive building that market 250 miles up and expanding that definition of global. And these are really exciting projects. I'm putting all 3 of them here, but the 2 that are stem cell based, you've probably heard about our artificial retina project, which is a protein polymer, but also we're looking at stem cell translational medicine with UCSD Sanford Consortium for Regenerative Medicine, and then Cedars-Sinai looking at parameter setting studies for stem cell manufacturing. So these are all really exciting and part of the foundation that we're laying for these types of products with the, you know, opportunities to manufacture is setting up GMP capabilities for manufacturing as well. So to close, you know, we really are on the brink of returning to space, and this time it's going to be to stay. So I'm super excited to have the opportunity to work with all of the teams that we work with, the great science that's being done, for Space Tango to continue to contribute capabilities that are automated that support sophisticated science.